Thank you all so much for being here today. I wanted to tell you a little bit about myself before I got started. And so earlier today, I was trying to think back to how and when I first got interested in the brain and in learning and memory and so on. And as I was, so I could share with you, and as I was thinking back on that, I realized even further back, I was first interested and sort of fascinated by human biology in general. And sort of starting from embryology and how a fully formed baby emerges from the womb and sort of all the way to adulthood, where you have all these organ systems that work together so intricately, precisely, beautifully to make us the beings that we are. And yet there's such a tenuous line between health and disease. All of that was so fascinating to me. And so when I started college, I told my dad, who is here today, I said, I think I'm going to major in biology. I said, I think I'm going to major in biology, and this irked him, because being the computer science professor that he is, and sort of the mathy engineer type, he said, don't you think it's a little descriptive? Don't you want to study something a little more quantitative? And I said, no, I really like biology, and I think I can learn a lot. And so I forged ahead. And so every time I'd come back home, and there'd be family and friends who would come by and ask, what are you majoring in? And I would say biology, and he'd sort of lunge across the room and catch us just in time to interject and say, she's studying to be a doctor. That was, in his mind, so much more justifiable. So anyway, hopefully over the years, I sort of stuck through with the biology, and hopefully that investment is slowly starting to pay off for him. But to be fair, I did study the biology together with the math and the computer science and the medicine, and all of that together has really made the biology so much more powerful and so much more rewarding. So he had a point. But it wasn't until I started my MD Ph.D. training at Columbia University with Eric Kandel that I really developed the passion to study memory. And many of you may know Eric, but I was sort of endlessly inspired by his enthusiasm, creativity, rigor, to study science and to study memory. And it was also at the same time that I was seeing patients as a medical student, and I would see these patients with profound cognitive disturbances, memory loss, very young people, older people, and all of that had a very strong impact on me. And after I finished the graduate training, I had the fortune to move on to Stanford University to work with Carl Dijsselroth for my postdoctoral training, and that was really where I was able to learn and contribute to several of the technologies that you'll hear me talk about today. And so earlier this year, I joined the Rockefeller faculty, and I chose to start my lab here for a lot of reasons, but the number one reason, I think, being that I think more than any other university, Rockefeller is unparalleled in its support for their faculty, especially the young faculty, in allowing us to pursue high-impact science, whatever that might be. But it wasn't until I arrived here that I realized that it's really a blessing to be here. And not just to be doing the science here, but to be doing that while being embedded in a greater New York City intellectual community, where folks like you are inherently interested, curious, engaged, contributing to the science that happens here. And so partnering with you in this process of discovery is so much more, it makes this journey so much more rewarding to us. So thank you so much for being here and for your support. And I'll go ahead and get started. So I thought I would start today by sort of sharing with you a short video on Clive Wearing. He's an accomplished musician who all of a sudden developed total amnesia due to a brain infection. And so unfortunately, this resulted in him not being able to form or store memories for more than 30 seconds at a time, and then he would forget them. And so he sort of lives his life in these 30-second cycles. And here's a short video into his life.
Do you know that we're making a film about you?
No, that's news to me.
You're being filmed for a television program.
Oh, no. Oh, dear. Oh, I suppose I'd be posh, do I? Upper class.
Because you're very famous.
Oh, likely story. You're full of jokes, aren't you? Are you two laughing as well?
About 20 years ago, a film was made about you.
Oh, no.
Just after you became ill, called A Prisoner of Consciousness. And 20 years on, we're making a new film about you. Because millions of people watched the film and wanted to know how you were.
Good heavens.
So millions of people know you,
If Clive went out of the front door, unsupervised, unaccompanied, it would be like being separated from a spaceship. It would be like if you were spacewalking and the rope broke, he would have no way of getting back, ever.
Can he read a book?
No, because he can't remember the sentence before last.
Can he watch a film?
No. He'll watch the rugby or he'll watch the cricket. He won't know who's playing or what the score is, but each stroke, each try, is satisfying for him to watch.
So memory loss is really profoundly debilitating, but I wanted to show this to note that Clive is very personable. He has a great personality, a good sense of humor. He can still play music extraordinarily well. And so folks like him might seem otherwise normal, and yet there's this deep void in their lives, an inability to really connect with people and experiences. And it turns out that memory deficits really manifest in a great many ways in our lives. We tend to think of them as emanating in large part from dementias like Alzheimer's, but Clive, for instance, had a brain infection. Many of us may, as we age, as Rick discussed, have deficiencies, declining memory abilities, which in and of itself can be disabling or socially isolating. And then there are profound memory deficits that are associated with Alzheimer's, schizophrenia, epilepsy, stroke, and many other conditions. And I also wanted to mention that memories can be disabling when they're too strong as well. And when we have associations in our minds that are so strong that they persist despite severe and repeated negative consequences. So we really want to be balancing this spectrum so that we're lying in this healthy middle ground here. But regardless of what the cause of these memory deficits and where they're emanating from, the truth is that sufferers are really in states of confusion, emotional pain, anxiety, and depression. So what do we do about this? How can we best tackle this problem effectively? And why is now a particularly opportune time to make an impact? So many of our behavioral manifestations are a result in large part of the changes in our genes that lead to changes in our the activity of our neurons and neural circuits. And by neural circuits, I mean the connections from one brain region to another that allows one brain region to communicate with another brain region. So it's the changes in genes and neural circuits that give rise to our behavioral manifestations. And traditionally, to treat disorders, we've taken a pharmacological approach to target genes and gene targets, or we've taken a behavioral approach. So for instance, we might use statins to target the cholesterol pathway. We might use antidepressants to target the serotonin pathway. Or behaviorally, we might turn to lifestyle modifications, physical therapy, psychotherapy. But what I'd like to talk with you about today is taking a neural circuits approach to understanding and intervening effectively for the treatment of cognitive disorders. And this is because we're really in a neural circuits era. In the last 10 decades, we've seen an explosion, a transformation in what our technology is able to accomplish. In the sense that we can literally, as Rick mentioned earlier, we can literally watch and manipulate brain activity at high speeds, high resolutions during states of cognitive disturbance. And this really gives us an insight into the neural circuits that are responsible, that underlie or that are responsible for the observed memory deficits or the observed cognitive deficits. And these sorts of insights can be directly translated into the clinic for viable treatment options. So for instance, deep brain stimulation, DBS, is a relatively new neural circuits approach that has really come to prominence in the last decade or so. And in this procedure, there's electrodes that are implanted just under the skin that are able to target electrical activity to deep brain regions for the treatment of movement disorders such as Parkinson's, but increasingly becoming more promising for the treatment of mood and cognitive disorders as well. And I should mention that this procedure, while it's invasive, is becoming less and less invasive, and just in the past year, there's new technology to make this fully non-invasive, which looks promising. And so this technology, this methodology has been most successful so far in the treatment of Parkinson's disease, which is a disease that results from the death of dopamine neurons in a brain region called the substantia nigra. And so pharmacologically, we try to replenish the dopamine in this region by providing dopamine to the brain, but actually, it turns out a more effective approach is to use a DBS-based approach, which is trying to do the same thing, but because it's trying to elevate dopamine neurons as well, but it does so by directly activating the remaining surviving neurons in the substantia nigra to increase the dopamine levels there. And this turns out to be remarkably effective. And I'll show a patient video in just a second, but I want to make the point, you might think, well, this process sounds really crude. We're just flooding certain parts of the brain with electrical activity. How precise can this really be? And so it turns out that just by changing the location or the frequency, high frequency or low frequency of the stimulation parameters, you can specifically affect one symptom class, but not the other. And this allows a fair level of precision so that if a patient comes in showing one symptom class but not the other, you can target that pretty specifically. And so here's a patient who he's displaying the cardinal symptoms of Parkinson's, which most prominently is rigidity, a lack of fluidity of movements, bradykinesia, sort of a slowness in movements, and a very typical gait disturbance. So as he was walking, you may have observed that he had a wider stance, sort of shuffling along the floor, and this enormous difficulty turning, which can be very typical. And with just after a few months on DBS therapy, here's the same patient who looks like a completely different person. He's very comfortable, walks fluidly, with ease makes the turn. And it's really a remarkable change in quality of life, and many, many, many patients have really benefited from this sort of procedure. And it really offers promise to be able to take this sort of approach and use that for other sorts of disturbances, for the treatment of other disorders as well. And so I just want to make a quick point here, which is that what we've learned, and sort of counterintuitively, is that this doesn't work if you target directly the diseased region of interest. So if we target DBS stimulation to the substantia nigra itself, we're not seeing effective results in patients. But if we activate the inputs, the fibers coming into the substantia nigra, then we see these very nice results. And so this is something I wanted to just point out and that I'll circle back to at the end of the talk when I discuss DBS options for Alzheimer's patients. But there are now currently FDA-approved DBS options, not just for Parkinson's, but also for obsessive-compulsive disorder, chronic pain. And currently, there's ongoing large-scale clinical trials to test its efficacy for drug addiction, depression, and Alzheimer's, which look very promising. And so if we are to really use a neural circuits approach to understand and treat cognitive disorders, where do we start? What do we know about the neural circuits that are responsible for memory processing? And what do we still need to learn? So for a long time, we've known that the hippocampus is the part of the brain that's really crucial to form and store memories. And we know that from examples in patients, from patients that have had heroic lives of total amnesia and unbeknownst to them have really given their life to science, and we've learned a remarkable amount from them. And here, for instance, is a very well-known patient, patient H.M. Henry Malazan, who passed away recently, but his life was an incredible learning experience for the scientific community. He, in particular, had epilepsy, intractable epilepsy, where the focus of his epilepsy was in the hippocampus. So in order to treat the epilepsy, the hippocampi were removed bilaterally, and unfortunately, as a result, he lost his abilities to form new memories and to store them. But we learned two very important things from his life, if we were to boil them down into two things. One, I would say, is that he had anterograde amnesia. When the hippocampi were removed, he had anterograde amnesia, which is inability to form new memories and to store them. But it turned out that it wasn't deficits in all kinds of memories. It was only certain kinds of memories. So he had a deficit in episodic memory storage, which is the memory for places, people, things, facts. But his working memory was totally fine, and his procedural memory was totally fine. So working memory, as in, if you were to look up a phone number in a phone book, and then from the time that you look it up to the time you dial it, you can hold that number in your memory for those 10 seconds, and he can do that. And procedural memory being riding a bike, or drawing something that you already know about. And so these procedures that he's already learned, he could already do. And so it was the first insight into telling us that not all memories are the same, and not all memories are stored the same or look the same in the brain. And secondly, he had a form of retrograde amnesia called temporally graded retrograde amnesia, in the sense that he couldn't remember what happened a year ago, but he could remember pretty well what happened way back in his childhood. And that tells us that sort of the first indication that memories aren't static. They're sort of moving around in the brain, and that some of his memories had escaped the hippocampus, and that's why he was still able to remember them. And so when we think, and this is sort of perhaps one of the most enigmatic parts of memory that we're still trying to grapple with, is just how dynamic of a process it is. You know, it's easy for us to think that a memory gets put somewhere in the brain and it stays there. But it's really not that way. And you know, we can put memories in one place, and then it can go somewhere else to become consolidated into long-term memory. Then it can go somewhere, and we can use completely different circuits to retrieve that memory. And it turns out that every time we recall a memory, we change it ever so slightly based on the current circumstances. We kind of fill in more details, then we put it back in memory, and maybe it goes back somewhere else. And then when we retrieve it again, we change it ever so slightly. And so this memory is constantly evolving over time, and we'd love to keep track of it. And that's sort of the holy grail here is, can we track a memory from start to finish and know how it looks, what is the representation of it in the brain, its form, its structure, its function, and can we track it over time from start to finish? And so this is really the area we still need to learn and grow a lot in. And ideally, we'd love to do this in humans, but we now have very sophisticated technology to be able to do just that in animal models, in rodent systems. So here's an example of a setup that I currently have in my lab, where we have a mouse here who's sitting on this trackball. His head is restrained, but he's comfortable. He's moving freely on the trackball. He can move in 2 dimensions. He'll navigate, he'll groom, he'll do things that he usually does. And yet he's in this environment that's a virtual reality environment where we're kind of playing a video game for him, and he's interfacing with this video game. And through this game, and it's not just a visual environment, I should mention. There's audio, there's scent, odors, there's textures. So it's a full sensory experience for him. And as he's playing this game, he can walk into certain rooms, receive rewards. He can go into other rooms. He might get mild punishments. And so in this manner, we can have him form new memories, learn them, store them over time, retrieve them in various contexts and conditions. And while all that is happening, we're going to peer into the brain, into the part of the hippocampus in his brain, and watch single neurons light up in real time as he's doing this behavior. And what you're looking at here are individual neurons, and when they light up, we know that that neuron was activated. And we can track this in real time and say, well, he turned left, and that neuron lit up every time he turned left. Or he licked, and that neuron was responsible for his licking behavior. So we can try to track what the function of these neurons are based on what the animal is doing by mapping these at millisecond precision. And so this image that you're looking at here, there are hundreds of neurons in just this one slice. And I'm taking just one slice through the hippocampus, but typically we collect these videos in large volumes. So we're getting thousands of neurons in these volumes in the hippocampus. And these are all taken with a 2-photon microscope, which uses a really high-powered laser and really sophisticated optical system that usually can take up an entire room. And so not trivial instrumentation. And so I just want to point out that a lot of this work is really standing on the shoulders of giants. And so, you know, this whole system of imaging rodent brain while they navigated in virtual environments, this was all really pioneered and popularized by David Tank and his colleagues at Princeton University. But since then, a lot of people have been able to collectively sort of extend that technology in important ways. And I, for instance, was very fortunate to work with Carl Dijsselroth at Stanford University during my postdoc, where I really had the opportunity to learn and contribute to the development of these technologies and extend them in important ways. So for instance, can we image faster? Can we image higher resolution? Can we image larger fields of view? And importantly, as Rick also mentioned, can we not just observe this activity, but can we play in activity? Can we then manipulate activity in real time so that we can causally bias his behavior? We can make him move left when he really wanted to move right. You know? So do we really understand the circuit enough to know that we can then modify the behavior in a particular way? And so what kinds of things can rodents do in these virtual environments? So just to give you an example of this, here are some behaviors that rodents typically perform. And so I'm going to turn this game on. And this game, basically, the mouse is going to get put in the circular arena, and he has to find this goal platform. When he gets there, he gets a reward, like a juice or sucrose water or something like that. And that's easy for him, but then pretty soon we'll make it invisible. And then we'll say, you have to use these cues to try to figure out where that reward location was. And this is maybe similar to when if you're going back to visit your childhood home, and the neighborhood, your house isn't there, but your neighborhood looks roughly the same. Maybe there's a grocery store there or a neighbor's house. And using those global cues, you're able to figure out where your house actually was. And so here's looking at it from the mouse's perspective, and here's a visible platform. So he knows that he has to go find it to get a reward. And so he can kind of almost misses it, backs up, walks in there. But he can do it when it's visible. So next, we make it invisible. And he knows that there has to be this pink ball, a soccer ball, and then this blue cube in the middle. And sort of around this quadrant here is where he's going to get the reward. We orient him in a completely opposite direction. He's moving the wrong way initially, but he usually turns around and he pans like most mice do. Oh, there he sees the soccer ball, but he's kind of missing the other ball. So he kind of turns around again. Oh, there he sees the pink ball and then the sphere. So he knows he's in the right area. He slows down and he hits it. And so in this way, we're able to keep track of how many trials are some mice learning this faster, some mice learning this slower. What is their latency? What is their speed? How fast can they actually find it? How many trials do they need to do a certain number of correct choices, and so on. And here's another example where, sorry, I missed that. So here, there are 3 different rooms down here, and one of them he gets rewarded, one of them he gets mild aversive punishments, like air puffs to his face, and then the other one is totally neutral, so he might get juice in this one and just a neutral water reward in the other. And this gives us an indication. So he's starting off here in an aversive room, but pretty soon this is a rewarding room. So he stops, and he drinks and he licks in anticipation of reward. But then as soon as he gets put in an aversive room, he runs right through it, and he knows this is not a room I want to stay in. And then he'll sort of move to other rooms, and eventually he'll get to another reward room, he'll stop, and he'll start licking again. And so as all of this is happening, we're collecting these volumetric data sets where we can track individual neurons in real time, and as they're flashing, we try to map it back to what the animal is actually doing. And these turn out to be very large data sets, and we're sort of moving. This is one brain region, but we can now image multiple brain regions at the same time, larger fields of view at the same time. So these data sets for one mouse, for one session, can be tens to hundreds of gigabytes, just one file size. And so it's not a trivial process to computationally store and then post-process and extract meaningful information from these data sets. So just to give you a sense for how we might go about that, I've borrowed this video from our colleagues at the Howard Hughes Medical Institute at Genelia Farm, where I think it beautifully shows, here's a rodent cortex, and where they're imaging, all of these flashing lights are neurons from different regions. So you have hundreds of thousands of neurons here, and the difficulty here, we can tell what a neuron is and what a neuron isn't, but in order to teach a computer to put dots where neurons are, and there's all these other little white dots that could be flashing, but that's noise. We know that's noise because that doesn't look like a cell, and it's not round, and it's sort of long and wispy, and so we know that's noise, but it's not trivial to teach a computer, well, this is the cell, this is not a cell, keep track of that cell over time. Even though there's slight brain motion and the animal might move ever so slightly, you still have to keep track of that neuron and keep registering it and motion correcting it, and eventually, you can use this process to say, these are all the neurons that we have in our field of view, and over time, we can track exactly what their activity patterns look like. And then we time lock that to the behavior and sort of compare that to what the behavior looks like. So here's an example of 6 neurons from here, and what their activity patterns look like over time. And what we can say is, at these times, there were auditory tones that were played to the animal. And so this neuron that fires every time the tone is on, we can say that's a tone-responsive neuron. And this other neuron that fires every time a different room was entered, we can say, well, that neuron's keeping track of which room this mouse is in. And so this sort of is somewhat simplistic, but hopefully it gives you a sense for how this kind of information can be extracted from these complex data sets. So what have we really learned? And through all of this, what can we meaningfully learn about memory processes? Well, I thought what I would do is to take you through one story in the lab, and just through sort of the next 4 or 5 slides, a project from start to finish, how that looks and what we really learned from that. And so at the time that I started this study, which was a couple years ago, at that time there was only a handful of studies worldwide that had looked at such high resolution in the brain of a mouse while it was performing a memory-guided task from storage to retrieval, from storage to consolidation to retrieval. So I started with a relatively simple task. And in this task, this mouse, every time he comes in this room, he gets rewarded. And so in the future, when he comes back in this room, he anticipates that he's going to get rewarded, and he has this aha moment. He'll start licking in anticipation. It's sort of like if you got an invitation to a reception at President Rick Lifton's house, and if you've ever been there before, you know the food's excellent, and the desserts are great, and the champagne's amazing. And so the next time you get an invitation, you're anticipating a very rewarding experience. And so that's what this mouse is going through. And it turns out we see something very interesting. What we see, if we sort of look at what all the neurons are doing, and this is just a fraction of the neurons in this field of view, but they're all lining up. Their activities are all happening at the same time, regardless of what that neuron is actually encoding. So each one of these is an individual neuron, and this neuron might be encoding the odor, and this one the texture. And by the way, these aren't just visual contexts, they're full sensory contexts. And so each neuron might be encoding something different, but there's a point in time while it enters that context where all the neurons fire together, kind of suggesting that there's an aha moment where the animal could think, yeah, I know this context. I know what should be happening here. And importantly, we only see this after training. We don't see this before the animal has learned this association. And we don't see it also when it's learned it with a weak association, with something it doesn't care about as much. So, if Rick offered us, Hershey's as opposed to truffles, that's not going to generate this kind of activity. And so, and so if you were to average all of this data together into these blue activity maps, you notice that there's these synchronous events. Here's one, here's another one. So when it's retrieving this memory, there's a lot of synchronous activity, and it's happening multiple times. But importantly, every time the synchronous activity happens, there's a small group of neurons that fire reliably right before the synchronous event. So before this one, this fires. Before this one, we have this firing, and before this one, these same group of neurons fire again. And if we go back and look to see how these neurons are all organized and connected to each other, we see that, and so this is each dot here is a neuron in our field of view, and each line is a connection when those neurons are actually talking to each other and functionally connected. And we find that these neurons in red are acting like hub neurons in the sense that when they fire, we get these huge synchronous bursts of activity. And this was really interesting to us because it doesn't exist before training. It doesn't exist for weak memories, but it does when you form a strong memory. You sort of pattern these memories in an interesting way such that activation of just a few neurons alone is going to lead to activation of hundreds of other neurons following it. So just to state that in another way, what we've learned is that a set of neurons can be sort of disorganized before a memory occurs, before a learning situation happens. But then after you've learned, you get a patterned network with the emergence of these hub neurons that are highly connected to the rest of the network. Importantly, these hubs only emerge after strong memories, not weak memories, and they act as conduits to recruit the rest of the memory representation. So they're just kind of priming you to then say, yeah, look, we're going to get all of these guys, which then is what is recruiting the rest of the memory representation. So why is this important? We've discovered this patterning, this hub-like activity. What does that really tell us about the workings of how memories are formed or stored in the brain? Well, it's providing new insights into how memories are patterned and organized. So for instance, first of all, it tells us which neurons are important, which ones we need to preserve, and which ones might be dispensable. So if this was a memory, these are the hub neurons. We want to keep those. Because we got rid of those hub neurons, we might not be able to get access to that memory and retrieve this process. These other neurons, they may be holding details of the memory, but they're more dispensable. We could get rid of those, and we'd still, in large part, know most of the memory and what that was about. So that's kind of like, for the New Yorkers here, that's kind of like the subway system. If Penn Station or Times Square went down, we'd know that would really disrupt traffic in New York and the flow of information. But if there was a small station in Washington Heights that went down, that wouldn't really affect the entire flow of information as much. And so much in the same way, we think memories can pattern themselves in this way so that they can keep track of which neurons are sort of the most important. And secondly, it provides a mechanism for how a single but important cue can rapidly lead to the retrieval of a full, rich, detailed memory. And so just as an example, if you had a significant other from the past, and you know, let's say they used a jasmine scent, and then several years later, you're walking by a jasmine tree, that entire flood of memories are going to come rushing back to you in almost an instant, you know, about that person, how they looked and how they acted and how they laughed and what they wore, all of these details just from the scent of that jasmine and that tree. And so it could be that these hub neurons are encoding the most salient features of your memory, and when you activate that, you're able to bring back the entire rest of that memory representation. And so we know that the hubs are really there in the hippocampus, but the question next is, who's accessing those hubs? When we want to retrieve that memory, what part of the brain is saying, yeah, let's go grab the hub neurons in the hippocampus, let's try to get to the rest of the memory. And so the truth is, much of the field has really focused on how we store memories. And that's why there's been a large focus on understanding the hippocampus. But what we know almost nothing, what we know very, very little about is how we perform memory retrieval. And that's in some ways more relevant to a lot of our disease processes. For instance, Alzheimer's, more and more we understand, is a retrieval deficit, not necessarily a storage deficit. That memory is there somewhere, and it might come back not at the right time, not when you actually quite need it. And so retrieval is a problem it's still quite enigmatic to us. And so we wanted to make some headway and to understand, other than the hippocampus, what else is being recruited during retrieval? And sort of said another way, what different brain regions are talking to the hippocampus, trying to grab and retrieve memories from the hippocampus adaptively in the right context? And so to do that, we injected tracers in the hippocampus. This is the hippocampus, where the hub neurons were. So I injected tracers here, really high sensitive tracers, that wherever you put them in the brain, they'll flow backwards. And so it's a way of finding the inputs to any brain region. And what we find is if we trace the bundles back, fiber bundles, it's largely labeling this part of the brain in the front of our cortex called the prefrontal cortex. And so what we did was discover a new pathway that had not been characterized before, where there's a direct connection from the front of our brains to the hippocampus. And this was actually really important and really striking to us, because while other inputs to the hippocampus had been known and characterized to some extent, this had been hypothesized to exist, and for a long time, people felt that such a connection would be an ideal retrieval circuit, because the prefrontal cortex is ideally positioned to integrate sensory information, cognitive information, emotional information. So all of that information comes together and is used to then say, okay, at this time, at this context, we're going to go in and grab this memory in the hippocampus. So it's thought to have been an ideal retrieval circuit, and yet it hadn't been really described or characterized before. So we wanted to understand this further and ask, what is it doing? Is it really a retrieval circuit? And so to do that, we turned to another technology called optogenetics. And this also is a technology that I was first exposed to in my postdoctoral work at Stanford, where Carl really was one of the pioneers of this technology. And the idea behind this technology is that you can make certain neurons, engineer them to be light sensitive. So we can put certain molecules in there, in those neurons, and then when we shine light, those neurons will become activated. So no need to put electrodes in the brain. This is all light-based. And just by shining light, and just by putting these molecules only in some neurons, only in this pathway, but not any other pathway in the brain, when we then shine light, only this pathway will be activated. And so now what I'm doing to this mouse, I want to test, can I actually retrieve a memory? Can I have the mouse retrieve a memory just by activating this circuit? Is it a retrieval circuit? So what happened with this mouse, the day before, I've trained him, I've put him in a context, and I've given him very mild shocks that made him uncomfortable. And so the next time I put him in that context, he might freeze. So this is like if you were meeting President Rick Lifton, but in your tenure evaluation in a conference room. It's sort of mildly uncomfortable, and then if you were to go back in that conference room, you might even be nervous or slightly fearful. So this mouse, if I were to put him back in that same context, he would freeze, kind of in anticipation of those mild shocks. But this time, I've put him in an entirely new context. This is the next day. He's in a different context. He's running around freely. He's exploring as he normally would. He even explores all these corners. Sometimes he grooms. He walks around. But pretty soon, the light is going to come on. This is all in time. See, the light has just come on. And that means I'm activating the circuit. And as soon as the light comes on, he gets into this hunched posture, and he starts freezing. And the light's gone off now, but he takes a little bit of time to sort of get back to what he was doing. And so he sort of runs around a little bit, but then he kind of goes back, and he goes back to that same spot as if that memory is still reverberating in his mind. And he's still sort of freezing. And this is something that happens across all mice that we tested. Very, very reproducible behavior. And so now he's slowly getting back to his normal routine, what he was doing, exploring, grooming, and the lights come on again. Uh-oh, I wanted you to see that part, but anyway, the lights come on again, and let me see if I can speed that up. There we go. So the light comes on again the second time, and he's in the middle of grooming, and he starts freezing again. So this, when I saw this the first time, really blew my mind. This was incredible, and really the power of this technology, and also just observing behavior. Behavior is so incredible that it's so stereotyped across every single mouse. We can make every single mouse do this, and so. But then there was that voice in me that said, "This isn't real." And many of you might be thinking the same thing. And this mouse is maybe he's just scared of the light. You're not actually bringing back a memory, or maybe you're just making him freeze. That's not because he's thinking of the memory. You just made him stop because you're affecting their motor system. So I went through systematically all of these controls, and I said, this only, and I found that this only works when that light-sensitive molecule is there. So when that light-sensitive molecule isn't there and you shine the light, the animal won't freeze, run right through the light. So he's not scared of the light. And it only works when he's trained on a particular memory. So if I just took him from the home cage and I shine light to activate this circuit, he won't freeze. He'll run right through the light. So it's not that I'm just activating a motor circuit that makes him freeze. It's only when I've given him this particular memory that's associated with a freezing response. And then we could say, yeah, well, maybe it's any circuit that's accessing the hippocampus. We know there's memory stored there, and if we just drove a bunch of electrical activity into the hippocampus, we'd be able to pull back a memory. But it turns out if you drive other inputs to the hippocampus, you don't get that response. So it was something very specific to this circuit that caused the retrieval of this memory. And furthermore, if you inhibit this pathway, the mouse cannot perform a memory retrieval that it normally would be able to do otherwise. We see an impairment in the strength and latency of that retrieval process. And so this was really striking. And so we took this one step further, and we said, okay, we think this is a retrieval circuit, and it's accessing stored memories in the hippocampus, but is it accessing those hub neurons? And is that the way that it's really able to access the entire memory representation? So to do that, we're doing the same thing. We're going to activate this projection again, but now we're going to image these neurons in the hippocampus at the same time. And we're going to ask which neurons are being recruited when this pathway is active. So here's the hippocampus imaging activity, and you'll see pretty soon a red light come on, and that's when I'm activating this pathway. And slowly, you'll see recruitment of more and more neurons in this field of view. And if we statistically, quantitatively pull out the neurons that actually get recruited by this pathway, we see that they were actually preferentially the hub neurons of that memory representation that get recruited first, and those lead to the recruitment of the rest of the memory representation. So to summarize this whole thing, this has really been a study or a foray into understanding a memory retrieval circuit. And we've discovered a new circuit that hadn't been described before from the prefrontal cortex to the hippocampus that is necessary and sufficient to retrieve certain forms of memory. And how does it do that? Well, one mechanism that we suggest is we think it's preferentially recruiting these hub neurons that are part of that memory representation, and as soon as the hubs get recruited, they recruit the rest of the memory representation, and the whole richness of that detailed experience comes back to you. And so, again, this idea that our memories can be stored that way in these hub representations are important for perhaps three main reasons. One is that it provides compression in some way, much the same way you'd sort of zip your files on a computer, sort of compress them. You know, we're allocating less real estate to every memory representation. We're saying the same memory, there's like hundreds of neurons involved, we're going to say only 3 or 4 of them are really, really central to that memory representation. And it also provides a measure of stability. And so you can destroy many of these other neurons, but most of the integrity of your representation is still stable as long as you're able to preserve your hub neurons. And finally, an ability to rapidly recall the memory. If you were to somehow access these hubs, you can right away get to the rest of that memory representation. And so just coming back sort of full circle a little bit to what we talked about with memories being on a spectrum, you know, if you can think of, you know, in Alzheimer's, it could be that these hubs get preferentially affected and you start losing memories not memories themselves, but your ability to retrieve that memory more rapidly than you otherwise would be able to do. Or in the case of, say, PTSD, maybe you have too many of those hub neurons and you start retrieving that memory inappropriately or in inappropriate contexts. And so we're sort of studying these avenues and sort of and that sort of is an ongoing effort in the lab. But I wanted to come back full circle and sort of ask, what is the relevance to humans? So all that I've talked about. And so this new prefrontal to hippocampus projection, the hubs that are there in the hippocampus, so we're working with some folks to figure out if there are analogous circuits in humans and if there are analogous processes that could be important for retrieval. But in the meantime, I wanted to point out that similar to prefrontal to hippocampus connection, there are other connections, for instance, the fornix to hippocampus connection, that people have been looking at in terms of a DBS, deep brain stimulation protocol. If you were to activate that circuit, is that going to improve memory in, say, Alzheimer's patients? So there have been large-scale clinical trials, phase 2 clinical trial that just ended last year, which showed promise. In patients over 65 years of age, there was a significant memory enhancement just by activating this circuit by DBS. It wasn't a full enhancement, but there was a partial enhancement that was significant. And similarly, there's another circuit, the entorhinal cortex to the hippocampus. These aren't clinical trials, but they're case studies. But when those are activated, you see enhancements in patients in memory performance. And so the interesting part is, importantly, DBS directly to the hippocampus has not been effective, in much the same way that in Parkinson's, directly activating the substantia nigra is not important, or is not effective. And here as well. And that also could tell us something about how, you know, targeting dedicated retrieval circuits may be much more valuable than targeting a storage circuit by itself. And so, you know, we do need more research to make these efforts more reliable, more robust, more suitable for the larger population. But we can get there if we understand the optimal targets. Maybe these aren't the most optimal targets, and we can target a different circuit that would be perhaps even more optimal. We can figure out what is the optimal timing of intervention. Should we do the DBS stimulation during learning, during storage, during retrieval, chronically? What is the timing of when to intervene? And what are the optimal stimulation parameters that can really get us there? So I'd like to end there, and I'd like to first and foremost thank you for really taking the effort to come here and for your patience through the talk. And I just want to thank the folks that have really made all of this work possible through their fundings and contribution. I want to thank my mentors who have really made all the difference, my past collaborators, and my current lab members who are seen here. It's really quite a talented bunch of graduate and postdoctoral scholars who make for a bright future, promising many discoveries ahead. Thank you.